Hello and welcome everybody. Welcome to the webinar this afternoon. Well, this afternoon, if you're on the same time zone as us in Stockholm. It's a pleasure to see so many of you coming in right now. While we're just waiting for everyone to come in and get settled and get listening, I wonder if we could just test the technical elements of the presentation, and just to make sure that you can hear me okay, to make sure that you can see everything. Why don't you go down to the chat and just type in, just to let me know where you're actually calling in from today, where you're listening in from. It'd be really interesting to know. And also to make sure that you see and work out where the chat function is and where you can ask questions as the talk is going on. So I'll just open up the chat myself there to see, so I can see when the replies start coming in. Also, of course, if there's any technical problems, you can let us know there. We're monitoring this chat all the way through this presentation, so we can kind of see what's going on. Who have we got? We've got London, British Library in London, Linköping, Hampshire, Stockholm. We've got some good representation between the UK and Sweden today. Newcastle upon Tyne, fantastic. Well, as I said, it's a huge pleasure to have you all with us today. My name is BILLITON from Ebba Biotech. Who are we? Well, we're a Swedish company that started in 2015 based on interdisciplinary collaborations between lots of different scientific fields. Our products are called optotracers, and these are fluorescent tracer molecules for labeling different macromolecular components. And that's exactly what we're going to talk about today. We're going to be talking about Amytracker. And without further ado, it is my great pleasure to introduce our speaker today. I'd like to introduce distinguished Professor Resia Pretorius from the University of Stellenbosch. She works within blood coagulation and circulatory inflammatory markers, and how they affect a lot of different diseases, from type 2 diabetes, rheumatoid arthritis, psoriasis, neuro-inflammatory conditions, and dare I say, COVID. She heads the Department of Physiological Sciences and has, through her career, amassed a whopping close to 200 publications. I'm extremely excited to listen to what she has to say, and I'd really like to join everyone in welcoming her to the Zoom stage now. Over to you, Resia. Thank you very much for being with us today.
Hello, everyone. Today, I will be sharing with you our latest research on clotting pathologies and how we use fluorescent markers, including those from Ebba, in various diseases. I will also spotlight our recent findings in COVID-19 and non-COVID, also known as post-acute sequelae of COVID-19, or PASC. I would just like to acknowledge that the work that is presented here, particularly the acute and long COVID research, was done with both clinical and research collaborators. I would just like to point out Professor Douglas B. Kell from the University of Liverpool, and he's also attending the webinar. And then my clinical collaborators, hematologists, and also Stellenbosch biochemists, and Dr. Chantal Venter, who is a physiologist and helped me with all my microscopy work. And then ethical approval was given by the Ethics Committee of Stellenbosch University and Mediclinic Private Hospital Group, approved sample collection at their facilities. My lab and our collaborators from various labs over the world have been identifying inflammatory molecules in circulation that might be involved in and drive pathological clotting. And we have also focused our research endeavors on studying the effects of increased circulating inflammatory molecules and how they interact with cells of the hematological system. We focus in particular on platelets and red blood cells, as well as on the clotting protein, fibrinogen. We are also interested in identifying novel inflammatory molecules that might play a role in the persistent symptoms of long COVID. Over the years, we have published numerous papers that show the importance of inflammatory molecules in circulation and their role in abnormal clotting. I'm particularly interested in platelet signaling and the role in abnormal clotting. Platelets in circulation play a critical role in healthy blood clotting. However, they can become overstimulated and can drive pathological blood clotting if there are inflammatory molecules in circulation. This can also happen in the presence of viral infections, where platelets can act as important signaling entities. There is also a complex relationship between receptors on platelets and endothelial cells, where circulating biomarkers may bind to. As we all know, damaged endothelial cells are also hallmarks of cardiovascular disease. We are also interested in P-selectin, a biomarker inside platelets and endothelial cells, but that can also act as adhesion receptors on the platelet membrane itself, or as a soluble biomarker in circulation. The clotting protein fibrinogen is also one of the molecules we have studied, as it plays an important role in clotting and then also in pathological clotting. It can also act as a circulating inflammatory biomarker that can bind to various receptors on platelets. Lately, the importance of damaged endothelial cells, platelet hyperactivation, and aberrant clotting has also been recognized in COVID-19. In the context of COVID-19, platelets are central in immune activation and general coagulation pathology. Although initially thought to be a typical viral pneumonia with acute disseminated intravascular coagulopathy, or DIC, it is now being accepted that COVID-19 is primarily an endothelial disease and also a vascular disease. Endothelopathy plays a significant role in the severity of COVID-19. Endothelopathy is also known to be significantly linked to coagulopathies, as it activates microthrombotic pathways and initiates microthrombogenesis, leading to endothelopathy-associated intravascular microthromby. It is now therefore well accepted that in addition to respiratory disease, cardiovascular and clotting complications are key threats in COVID-19. Myself and Douglas Kell have also had a keen interest in the structural changes in the biochemistry of fibrinogen and how circulating inflammatory biomarkers might directly bind to this clotting protein to induce protein misfolding. If you focus your attention to the two cartoons shown in Figure A, on the left is a healthy protein structure, which has many alpha coils and few beta sheets. However, in the presence of inflammation, oxidative stress, and circulating biomarkers, this structure changes where the alpha coils untwist into beta sheets, as seen in A in the cartoon on the right. In plasma clots from healthy individuals, these clots have more alpha coils and have the ultra structure shown in Figure B. Various inflammatory conditions, including cardiovascular disease and diabetes, have a clot structure similar, as shown in Figure C. So with our knowledge of platelets and abnormal plasma protein structure in various inflammatory and cardiovascular conditions last year, we focused our attention to COVID-19. You might be familiar with the clotting pathways. Normal clotting not only involves the enzymatic pathway, culminating in the formation of fibrin, but also platelet activation. Also important is the process of fibrinolysis, or breaking down of blood clots when it forms. Here is a cartoon that depicts the progression and severity of the acute COVID-19 disease and what happens to fibrinogen. In a healthy individual, fibrinogen is soluble. The structure of soluble fibrinogen changes to form fibrin-containing microclots, and the action of thrombin and the formation of D-dimer are pathological, where D-dimer levels are increased in very ill patients. Now, with this in mind, we looked at platelet-poor plasma and exposed it to our fluorescent markers theoflavin T, which binds to open hydrophobic areas on damaged protein. This marker was first used to identify amyloid protein in brains of Alzheimer's disease. In 2014, myself and Douglas Dakel discovered that theoflavin T also binds to misfolded fibrinogen and fibrin protein during pathological blood clotting. We can also use scanning electron microscopy. And here you can see spontaneously formed microclots in whole blood from acute COVID-19 patients. Note the yellow arrows. We also looked at acute COVID blood samples, as I mentioned, using scanning electron microscopy. And in the micrographs on the left, you can see platelets are hyperactivated and clumped with numerous extensive pseudopodia formation. They've also shed their content and extensive membrane damage can be seen. Note also the yellow arrows. In the micrographs on the right, platelet and red blood cell interactions can be seen. These samples were taken from treatment-naive acute COVID-19 patients that were hospitalized. A smear was made from a citrated blood sample after collection. So using this information, we also recently suggested that microclot and platelets can be graded using fluorescence, and we based our system on various stages of activation of severity of coagulation. This publication is currently online. Stage 1 is shown as minimal microclot formation in healthy and controlled plasma samples, which progress to the presence of severe microclotting in stage 4. Here you can see platelet-poor plasma exposed to theoflavin T. The bottom row represents examples of stage 4 microclots using bright field microscopy in A, fluorescence microscopy in B, and an overlay of fluorescence and bright field microscopy in C. It's important to know that clots may also be detected without fluorescence, which is the autofluorescence, and a little bit more about that later. Fluorescence microscopy examples of the different stages of platelet activation was also done, and spreading is shown here. Stage 1 are examples of platelets from healthy or control samples with minimally activated platelets. In healthy blood, platelets are small, round cellular entities with a few pseudopodia, visible due to contact activation. Platelet pathologies progress to egg-shaped platelets, indicative of spreading and the beginning of clumping as seen in stage 4. Here you can see fluorescence microscopy examples of the different stages of platelet clumping. No clumping occur in a typical healthy or control sample. This stage is graded as stage 1, where no figures are shown due to the absence of platelet clumping in healthy blood samples. As platelet pathology progresses, so does the clumping severity, which is shown in stage 2 to 4. We also suggested that this can be used in a numerical scoring system. Here we can see microclot formation in platelet-poor plasma in controls versus diabetes and acute COVID-19, demarcated again with our green signal from theoflavin T. We found small areas of nearly undetectable abnormal clotlets present in plasma of healthy individuals, as can be seen in the micrographs on the left. More abnormal microclots were noted in plasma of individuals with diabetes. This is in line with our knowledge and previous publications of the presence of hypercoagulation in type 2 diabetes, which is indeed well known. What was, however, quite significant to us was the extent of the abnormal microclots in acute COVID-19 plasma. You can see the micrographs on patients with acute COVID-19 on the right. Also note the scale of 10 micrometer at the bottom of the micrographs. Coming now to some of our Ebba work and Amytrackers. Here we show plasma clots from acute COVID-19 patients using Amytracker 480 and THT, which is the green signal. Here I wish to also mention that fibrinogen is notorious to show autofluorescence in many of the wavelengths that you can detect signal from. Our signal settings, and if interested in basically mean fluorescent intensity, it is crucial to determine autofluorescent signal first. For the microclot research, we are more interested in the area of microclots than in the mean fluorescent intensity, but some of our other research and published publications, we did take mean fluorescent intensity into account. We previously studied plasma clots in other conditions. Here is an example from our Parkinson's disease research papers. These clots were formed by adding thrombin to platelet-poor plasma from patients with Parkinson's disease and also those that are not diseased or healthy control samples. In these confocal micrographs, THT, Amytracker 480, Amytracker 680 was used to identify anomalous clotting. Control samples have very little THT or Amytracker signal, as would be expected and seen on the first row. We have also previously used a purified FITC fibrinogen model, where we also added thrombin to create extensive fibrin nets. An example of such a clot is seen in A, the top left micrograph. In micrograph B, a FITC fibrin clot is seen after we added serum amyloid A, a well-known acute phase inflammatory molecule. The dense and bright green signal shows clot pathology and hypercoagulability. The two bottom micrographs show purified fibrin clots using this model with added serum amyloid A and Amytracker 680. Note the red signal. Here is a 2020 paper where we showed fibrin clots prepared by adding thrombin to platelet-poor plasma from colorectal patients. We converted the images to grayscale. Amytracker 630 and autofluorescent signal was detected and taken into account in this paper. We also looked at tissue biopsies from colorectal patients using Amytracker 630 as our amyloid-specific marker. For this presentation, I just increased the micrograph fluorescent intensity to show the areas of Amytracker marker a bit better on the screen. In column A, you can see confocal micrographs of colorectal tissue biopsies from healthy individuals, and column B, biopsies from colorectal cancer patients. A scatter plot is shown where mean fluorescent intensities of healthy versus tumor tissue were analyzed, also with AMY630 using as our mean fluorescent intensity marker. Now a bit more on our latest research on long COVID or PASC. Across the globe, a subset of patients that have recovered from acute COVID-19 experience a wide range of persistent symptoms that do not resolve over many months. This new syndrome of COVID-19 phenotype is commonly termed long COVID. Some of the main lingering symptoms include recurring fatigue or muscle weakness, also being out of breath, and patients complaining of sleep difficulties, anxiety, or depression. Other common complaints are forgetfulness and also struggling with concentration. About 150 symptoms have been identified that is linked to long COVID. Globally and in South Africa, we are only now beginning to see the actual impact of long COVID on individuals, but not only on individuals, also on the economy. The Long COVID Alliance in the USA reported that between 25 and 35% of patients struggle with lingering long COVID symptoms. We believe that these numbers will not be different in South Africa. An important consideration is that the debilitating side effects of the symptoms may have a significant effect on our workforce and healthcare resources. Some people have reported that they even struggle to perform work at the same level as before their acute COVID-19 illness. So now back to the research and what we found. We noted significant platelet hyperactivation and clumping in the blood of individuals with long COVID. In the micrographs on the left, you can see representative platelet micrographs of healthy individuals, and on the right, platelet hyperactivation and clumping, which are visible in blood of the long COVID patients. Here are examples of microclots in THT and Amytracker 480 as our choices of markers. We also planned an experiment where we looked at proteomics of healthy plasma versus type 2 diabetes, acute COVID-19, and long COVID. We added diabetes samples as we know that people suffering from diabetes are more prone to severe COVID-19 symptoms. For proteomics analysis, we prepared platelets, poor plasma from citrated blood, followed by a first trypsin digestion step. To our surprise, we found a visible deposit at the bottom of the Eppendorf tube in the acute COVID-19 and long COVID samples, but not in the diabetes samples and also not in the control samples. Plasma proteins, therefore, were fully digested in control and diabetes samples, suggesting that trypsin could degrade all, or then at least most of the plasma proteins in both controls and diabetes. For proteomics, the sample supernatant was filtered with a C18 filter, and we also followed a second trypsin digestion protocol to solubilize the undigested pellet deposit. We analyzed 1 microgram protein of the supernatant and also the equivalent from the digested pellet deposit. We then viewed the unfiltered supernatant after the first trypsinization step using fluorescence microscopy. We found that plasma of patients with type 2 diabetes and plasma from healthy patients digested fully after a first digestion step. See the micrographs on the top left. No signal of fluorescence was noted. In contrast, microclots in plasma from patients with both acute COVID-19 and those with long COVID did not fully digest with the first trypsinization step as mentioned. Here we show these microclots that are still present in plasma after the first trypsinization digestion step. After the second step of digestion, we could then digest the microclots in the plasma from both acute COVID-19 and long COVID patients. We detected various inflammatory molecules that were substantially increased inside the now digested microclots of patients of acute COVID-19 and long COVID versus the equivalent volume of fully digested fluid in the control sample. Of particular interest was a substantial increase of alpha-2 antiplasmin in the various fibrinogen chains that were trapped in the solubilized fibrinolytic resistant microclots. Of interest was also the presence of serum amyloid A, an acute phase protein. As mentioned previously, serum amyloid A is a well-known molecule to be dysregulated in inflammatory conditions, including diabetes. This also is well known from our previous research, where we noted that serum amyloid A can also incorporate itself into fibrin clots, as shown in some of the previous slides. Briefly returning to alpha-2 antiplasmin, this diagram shows the clotting and lysis pathways we are all familiar with. The molecule alpha-2 antiplasmin prevent clots to lyse, and if it is present in plasma of long COVID patients, it might be the cause of the persistent microclots in circulation that do not break down via the normal lytic process. We have recently started a collaboration with Dr. Beate Jaeger from Germany, who has been treating numerous long COVID patients with great success. Together with Dr. Jaeger, we have been gathering like-minded researchers and well-respected clinicians all over the world. Dr. Asad Khan is one of our collaborators, and he is a respiratory physician from Manchester University Hospitals, currently receiving treatment for long COVID under Dr. Jaeger. Hopefully the results that we will have gathered over the past week in Germany will give clinicians confidence in anticoagulation and apheresis to act as a proof of concept for larger multi-center studies. Dr. Jaeger uses the apheresis system, so a bit more on this system. It's called HELP apheresis, and it is a technology now used for microclot removal from circulation of patients with long COVID. From a global perspective, more than 3,000 patients are treated with HELP apheresis every year. In this context, HELP apheresis is well established and more than 30 years of experience, and nearly 500,000 treatments worldwide. HELP is a highly selective kind of therapy. Atherosclerotically effective risk factors such as lipoprotein A, fibrinogen, and inflammatory factors are removed additionally and apart from LDL cholesterol. And lately, Dr. Jaeger's team have also been using this treatment, as I mentioned, for long COVID patients. Here is an example of a clot or clots inside the filter from a patient with long COVID that has been on the system. You can see the microclots that has been filtered from the patient, and it is really visible by the BI. Here is an example of abnormal blood clotting in blood from a long COVID patient. You can see the clot that is lying on the little paper at the bottom that the person is holding. We have just gathered data, as I mentioned, from patients before and after treatment in Germany. And there we also used Amytracker markers together with our trusted THT. Just a few of our collaborators on long-COVID, and we hope to report back soon with more interesting results where we used Ebba's Amytrackers. We have been using Ebba's Amytrackers since 2017, and hopefully we will still have many publications with these really cool markers. I end this talk with my website, where you can find all my publications on the website.